Stem cell therapy versus gene therapy in Parkinson's disease, a comparative analysis. Hi everyone, my name is Alavya, and my mentor for this project was Dr. Deblina Day. So I'm going to be talking about an overview of Parkinson's disease, some current pharmaceutical treatments, stem cell therapies, gene therapies, and my conclusions. So Parkinson's disease, first named by James Parkinson in 1817, is a progressive disorder of the nervous system. Some symptoms patients experience include tremors, bradykinesia, loss of automatic movements, and anxiety. Around 90% of these cases are sporadic, which means that there is no known genetic cause. Parkinson's disease is usually classified into 5 stages, stage 1 and 2 being higher risk of falls and tremors, which don't necessarily interfere with daily activities. Stage 3 and 4 are where things get a little bit more serious and help is necessary and independent living becomes a challenge. By stage 5, unfortunately, most patients are unable to stand, walk, eat, or swallow. PD affects almost 10 million people worldwide, which is why it's almost incredibly necessary to find a cure and to look at different treatments. Around 0.5 to 1% of these cases include patients that are 65 to 69 years old. And around 1 to 3% of these patients include patients that are 80 plus years old. The reason why I picked to study Parkinson's disease and the reason why it's so relevant is because there is an expected 30% increase in cases by 2030, and yet by now we have no clear cause for Parkinson's disease, no specific cure, and several treatments are still being debated. So some causes of Parkinson's disease have been attributed. Some factors have been attributed to Parkinson's disease, and it's primarily caused by the degeneration of dopamine-producing nerve cells. Some of these factors are genetic or environmental toxins or protein misfolding. Genetic can be attributed to around 10 to 15% of cases, and that's mostly concerned with Lewy body accumulation. Environmental toxins like pesticide, herbicides, heavy metals, and alpha-synuclein misfolding have also been attributed to Parkinson's disease. So this is the basic pathway of dopamine, the regular one. So you wouldn't see this in a PD patient because their dopamine pathway is irregular. But basically, dopamine is released from the substantia nigra to the basal ganglia and then released. So these are some of the current pharmaceutical treatments for PD. And the reason why this is so important is because the side effects and the effectiveness of these drugs have told us why alternative treatments are necessary. Drugs like L-Dopa have side effects like dyskinesia, nausea, low blood pressure, and motor fluctuations. Dopamine agonists cause sleepiness, dizziness, and impulse control issues, and MAO-B inhibitors cause insomnia, nausea, and confusion. What's more is a lot of these drugs are only short-term effects, and they only really help early and mid-stage Parkinson patients. This is where stem cell therapy and gene therapy come into play. In 1958, stem cell therapy was first introduced, and it involves the use of stem cells to repair damaged tissues or transform into other cell types. A lot of these stem cells can be derived from bone marrow or embryo, and they're usually delivered by direct injection. Gene therapy, meanwhile, first introduced in 1972, involves the replacement, silencing, or correction of faulty genes. Delivery is usually through vectors, and it can cause different immune responses, which we'll talk about more. So beginning with stem cell therapy, this is just a general image and a reference of how some stem cells can be derived. You can see NSCs from fetal brain, pluripotent cells from blastocytes, dopaminergic neurons from fibroblasts, and MSCs from bone marrow. So HFVM tissue was one tissue actually that was being explored as a cure for Parkinson's disease, or more so a treatment. Human fetal ventral mesencephalic tissue has emerged proof that stem cell therapy can aid in the replacement of dead dopaminergic neurons. However, this tissue is taken from ventral midbrain of human fetuses. This means that it originates from aborted fetuses, and this brings a huge ethical concern when it comes to the use of HMVM tissue. While clinical trials do show promise, the ethical concerns really outweigh the promise that it shows. This is just a general graph of what some studies with HFVM tissue show, and it's basically a general reduction in UPDRS score, showing long-lasting relief of motor symptoms, but again, we cannot ignore the ethical concerns that comes with. Mesenchymal stem cells are another type of stem cell, and they have properties of self-generation, differentiation, and the abundance of MSCs and their non-tumorigenic properties add to why they're extremely promising. And to mention that they have no ethical concern that, I wouldn't say no, but less of an ethical concern that HFVMs bring. The paracrine actions of MSCs mediated by exosomes have actually also proved useful in treating a variety of diseases. And MSCs can also be derived from dental pulp stem cells, which show greater promise in colony formation than bone marrow-derived stem cells. iPSCs are another emerging source of stem cell therapy, and these are basically induced pluripotent stem cells. They were first discovered almost 10 years ago using mouse embryonic fibroblasts and the forced expression of Yamanaka factors. Along with MSCs, iPSCs also avoid ethical concerns, but they do pose significant risks of tumor formation. So I'll go back to this image, but basically iPSCs are formed from fibroblasts. In 2018, researchers in Kyoto, Japan conducted the first human trial involving iPSCs, and this was with the in vitro method. It involved fibroblasts taken from patients with PD, which are then reprogrammed to a pluripotent state using transcriptional factors. Here we can go back and see how these fibroblasts are dedifferentiated, then there's genome editing, differentiation, safety and quality checks, and then transplantation. So while this did show incredible successes, it's still very complex and risky. This is a general overview of a lot of the stem cell therapies. We talked about embryonic stem cells, which are FMVM, mesenchymal stem cells, induced pluripotent stem cells, and neural stem cells are also actually part of Parkinson's disease research. Neural stem cells release DA after transplantation, but they do have side effects like dyskinesia and tumor formation. All of this ineffectiveness and these side effects have led a lot more research going into gene therapy. So two main types of gene therapy that are very promising in Parkinson's disease is GDNF and AAV2 GAD gene therapy. So GDNF is a type of growth factor gene therapy, which means that it promotes the survival and growth of dopamine-producing neurons. This influences neurite sprouting and reduces PD symptoms in toxin models. Now, AAV2 GAD gene therapy works a different way by using the modified virus AAV2 to deliver the GAD genome. This helps release more GABA, which regulates brain activity. GDNF, however, is short-term, and it has had a few unsuccessful non-viral techniques, while AAV2 GAD gene therapy exhibited improved motor scores, which we'll talk about more. So this is an example of an AEDC study done by Dr. Chadwick and colleagues, and they used trials including 15 participants aged 40 to 70 with 5 or more years of diagnosed Parkinson's disease. With using cohorts with varying degrees of dosage, they found through a PET scan that cohort 1 with the lowest dose saw an increase of 13%, cohort 2 with medium dose, 56%, cohort 3, 79%. And because this study was done over 3 years, they also found that a lot of the groups with higher doses required a reduced levodopa equivalent dosage by 21 to 30% after 3 years. This is just a general graph so you can visually see how cohort 3 really improved. This is an example of an AAV-GAD study done by Nehammer and colleagues using the AAV2 viral vector in a double-blind sham-controlled trial. Using 45 Parkinson's disease patients as participants, this trial was done for 6 months as an open-label phase and used a unified Parkinson's disease rating scale. So Nehammer and colleagues found that the patients treated with the injection had much better motor scores at 12 months compared to the sham surgery group. And patients who received the AAV2 GAD gene therapy did not experience levodopa-induced dyskinesia. In total, I just want to talk about the differences between stem cell therapy and gene therapy. While stem cell therapy has had its successes, it's clearly ethically challenging and has risk of tumor formation. While gene therapy aims to target specific cell types and brain regions, it lowers risk factors and shows greater results, and is generally more ethical. At the end of my study, I just want to give a heartfelt thank you to my mentor, Dr. Deblina Day, for her patience, kindness, and support through the entire research process. Thank you so much.